Here are two USMLE style questions focused on the capsule of bacteria, which is typically made of polysaccharides and rarely proteins and provides protection against phagocytosis. Question 1. A 65-year-old man with a history of chronic obstructive pulmonary disease, COPD, presents with fever, cough, and purulent sputum. A sputum culture grows Streptococcus pneumoniae, which has a polysaccharide capsule. The capsule plays a critical role in the pathogenicity of this organism. What is the primary function of the polysaccharide capsule in Streptococcus pneumoniae? A. Mediate attachment to host cells. B. Inhibit bacterial protein synthesis. C. Enhance bacterial motility. D. Protect the bacterium from phagocytosis. E. Facilitate horizontal gene transfer. Correct answer. D. Protect the bacterium from phagocytosis. Explanation. D. Protect the bacterium from phagocytosis. The capsule is a protective layer that inhibits the host's immune cells, particularly macrophages and neutrophils, from recognizing and engulfing the bacterium, enhancing bacterial survival in the host. A. Mediate attachment to host cells. While pili fimbria are responsible for attachment, not the capsule. B. Inhibit bacterial protein synthesis. Not a function of the capsule. This is more relevant to antibiotics like tetracycline or chloramphenicol. C. Enhance bacterial motility. Motility is driven by flagella, not the capsule. E. Facilitate horizontal gene transfer. Horizontal gene transfer is facilitated by plasmids or pili, not the capsule. Question 2. A 42-year-old woman presents with a persistent fever and an enlarged tender lymph node. She is diagnosed with an infection caused by Haemophilus influenza, a bacterium that has a well-defined capsule made of polyribosylribitol phosphate, PRP. How does the capsule contribute to the virulence of Haemophilus influenza? A. Inhibition of bacterial protein synthesis. B. Protection from immune system recognition and phagocytosis. C. Enhancement of bacterial motility. D. Facilitation of bacterial adhesion to epithelial cells. E. Formation of biofilms on medical devices. Correct answer, B. Protection from immune system recognition and phagocytosis. Explanation, B. Protection from immune system recognition and phagocytosis. The capsule of Haemophilus influenzae prevents opsonization and phagocytosis by immune cells, helping the bacterium evade the immune system and persist in the host. A. Inhibition of bacterial protein synthesis, not related to the capsule, but could be relevant for antibiotics or specific toxins. C. Enhancement of bacterial motility. The capsule does not facilitate motility, which is usually due to flagella. D. Facilitation of bacterial adhesion to epithelial cells. Adhesion is primarily mediated by pili or other surface structures, not the capsule. E. Formation of biofilms on medical devices. Biofilm formation is associated with other bacterial factors, e.g. surface proteins, though capsules can contribute to biofilm formation indirectly by enhancing bacterial survival. Would you